Okay, hello everybody. So I guess we better start on time. So our subject today is going to be plant viruses, which are important to cover because, firstly, of their interest in, plant biology. But from a fundamental virology point of view, plant viruses are also interesting because they have particular characteristics that we don't find in animal viruses. So if we want to have a good general knowledge about virology, you can't just study animal viruses and think that you know everything. You have to cover plant viruses as well. So the first thing I'm going to talk about is, the types of viruses that we find in plants, not really taxonomy, it's a little bit of a grand statement. And then, so what are the specific problems that plant viruses face that they all have to solve in some kind of way, but we don't find in animal viruses. So one of them is how to infect the plant in the first place. And then for local spread within the infected plant and systemic dissemination, these are two different processes, and every plant virus has to have an efficient solution for both of these stages in the infection of a plant. So once we've done that, then we'll look at a few specific examples. Tobacco mosaic virus, it's the big example that everybody knows. Then another filamentous plus strand RNA virus, potivirus family. Then DNA viruses, the Gemini viruses. And then we'll end up looking at some types of replicative genomes that are quite specific to plants. So multipartite genomes, the presence of satellites, and we'll end up with viroids. So the first thing, what kind of viruses do we find in plants? So this was my fantastically historical slide. I must have prepared this about 15 years ago minimum. So what it's trying to represent is the kind of diversity within each of the Baltimore classes. So the size of the letter set is proportional to the number of different virus families within each Baltimore class. And then these are the types of particles that you find within those classes. So the two most prevalent types of virus genomes are double-stranded DNA and plus-stranded RNA. And we can understand this by thinking that these guys, their basic strategy is to look like the host cell DNA. And these guys, their basic strategy is to look like the host cell messenger RNA. And then they just elaborate around these ideas. So what about if we look at plant viruses? So in plant viruses, it looks like this. Plus-strand RNA viruses, still very, very frequent, and it's the most prevalent group in plant viruses. But the big hole here is double-stranded DNA viruses. There are no double-stranded DNA viruses that infect plants. And you might wonder, okay, so why is this the case? It's a really unusual thing because it's the most prevalent group in animal viruses and also in bacteriophage. So what seems to be the problem for these viruses is that they can't really transport their genomes from cell to cell to have this stage of efficient local spread between plant cells. So so that seems to be the step that is impossible for double-stranded DNA viruses. So there are single-stranded DNA viruses, the Gemini viridae, and we will see that these guys have got a very, very small genome. It's only about 3 kilobases in single-stranded DNA. And it's also thought that this step of transfer of the genome from one infected cell to another is what limits the genome size of these viruses. Well, it's one of the things anyway. So no double-stranded DNA viruses in plants, and kind of limited diversity in the families of the other Baltimore classes, except for the plus-stranded RNA viruses, which are very diverse. However, the things we do find associated with plant viral infections are a lot of satellites. So these are kind of mostly RNA molecules that can replicate when a helper virus is present. Okay, germinal viruses also have DNA satellites. So you get the infectious virus with a full genome, and then you, for some types of viruses, you also have like satellites that are co-infecting plants. And the other thing we find in plants, but not in animals, are viroids, which are a very, very strange biological object indeed. Okay, so what about these steps that are difficult for plants? The first one is infecting a new plant. And in fact, there's no direct infection of plants by viruses. So there's no specific receptor protein. So everything we talked about for animal viruses, the whole thing starts with a specific interaction between an attachment protein or glycoprotein on the virus and a receptor protein on the host cell. That's not true for plant viruses. So all, because that means, that the cuticle and the cellulosa cell wall seems to be an effective barrier to prevent viruses from infecting plant cells. Initially, anyway, to get the initial infection. So that means that almost all, well, not all, but almost all plant viruses are dependent on a vector in order to get into a susceptible host. So most plant viruses use insect vectors, which are either going to like chew up the leaves, or these will be insects that can feed on the sap of plants. So they'll be injecting virus particles directly into the plant vascular system. So two mechanisms of entry. There are plant viruses that use fungi as vectors, so specifically molds, which can insert they can attack susceptible plants by inserting their mycelium into the plant cells. And in some cases, they can also carry virus particles to invade the plant. Okay, so the vast majority of plant viruses are dependent on some kind of vector for entry. However, it's not true for absolutely all viruses. So there is vertical transmission. So sometimes you get viruses transmitted with the seeds. So, plants are infected from there as soon as the seeds germinate. And there are some important viruses like the tobacco mosaic virus, which, as I said, is the big classical example that everybody knows. This is a kind of unusual exception because there's no insect vector which is known. So it seems like the transmission is through agricultural practices. So of course, if you are pruning plants with pruning scissors, okay, and if you've got like 10 plants all in a row, one of them is infected, you'll cut off the leaves off the first plant or harvest them. If it's like tobacco, you're cutting them, and then you move to the next one. But the pruning scissors you have, they'll be infected, okay, with some virus. And when you do you cut off the leaves of the next plant, you can transmit the virus. So pruning also, what's the word in English for that? Yeah, when you have a graft of one plant on another plant. If you graft one branch onto another plant, onto another tree or something, then you connect their vascular system. So if the graft is infected, you can infect the new host. Okay, so this seems to be the main mechanism of transmission for tobacco mosaic virus. And sometimes you don't really need very much to transmit the infection. So for example, if you got tobacco mosaic virus, the leaves will have a high viral load, and you collect them, they'll be on your hands, and you go and collect them on another plant, just like rubbing this infectious material into the plant will be enough. Okay. So agricultural practices is a mechanism of transmission of viruses. Okay, but most plant viruses that are problematic are vector transmitted by insects that either just eat the leaves or are feeding on the sap. So whenever you get a specific infection by one plant virus that will infect a specific host, it will often be restricted also by a specific vector. So for example, these Gemini viruses up here, okay. Okay, now I should perhaps start by talking about the names here. So for the names, I've put like the kind of classical name of each of these plant viruses, and I've also added their systematic name. This is their official name from the ICTV. Now, the way the classical virus names work for plant viruses, okay, last letter, that's easy, that's for virus. The first letter was the host plant. Okay, so this is tomato, host tomato. CA, cassava, M for maize, T also tomato, B for BA for barley. Okay, so the first letter was for the host plant. V at the end is for virus, and everything in the middle is to do with the symptoms, the type of phytopathology that the infection provoked. So this one is tomato yellow leaf curl virus. This one is cassava mosaic virus. This is maize streak virus. And this is tomato spotted wilt virus. And this is barley, I think, yellow mosaic virus. Also, you have different names like yellow mosaic, yellow mottle. Sometimes it's difficult for me to remember. Okay, so host species and then something to do with the symptoms. And sometimes this comes through into the species name. Okay, so like TSWV now, Tospo, this is the genus, and it's got tomato maculae, so tomato and then the spots. Kind of is, the old name is kind of been translated into Latin here. Same kind of thing here. So Horde for barley and Lyceum for yellow. Okay, so these are the viruses. These are the host plants, and these are the associated vectors. So the countries or the regions that have a problem with a particular virus depend on where the host plant is cultivated and where the vector can survive. So for example, MSV is only a problem in sub-Saharan Africa. Okay, we got plenty of maize growing in Europe and in France, but we don't have a problem with this virus because the vector doesn't live here. And similarly, these viruses used to be, I believe, restricted to Africa, but since then, the vector has managed to colonize other continents because of the transport of agricultural, crops, harvest that are exported from one place to another. Within the crop, you often have the eggs or live insects. So the vectors got transmitted to new continents more recently. So cassava mosaic virus used to be restricted to Africa, but now you can also find it in India because the vector has managed to get there. What are the points here? So the same vector can transmit several viruses. Bemisia tabasi transmits more than one virus, and the same virus can infect several host plants sometimes. So the TSWV is kind of the champion of this. It can infect hundreds of different plant species. And the only thing that really restricts the spread is its dependence on a particular vector. So what is the other thing I want to point out about this is, okay, so tomato, cassava, maize. Where did these plants all come from originally? Think about maize, right? That's an easy one. Yeah. Yeah. South America, Latin America. Where did we first see the virus emerge? Often Africa or another tropical or subtropical region, but not Latin America. So what's the origin of these highly pathogenic infections? We've got a plant species from South America, and like the cassava or the manioc, same thing, right? We've got it, it's from South America, we've started to grow it in Africa. So it's not in its natural environment, and it's going to meet a lot of new viruses that are naturally infecting monocot plants in Africa. So you've got a kind of encounter between a virus and a new host. So when this happens, it's the same thing with zoonoses and animal viruses. Okay, you've got some kind of species barrier. So often the virus can't infect the host, nothing happens. But if the virus can infect the host, often you can get a highly pathogenic infection because there hasn't been any time for the host and the pathogen to co-evolve. So, you know, the host resistance mechanisms, all of these like manioc plants or the cassava plants, they're all trying to like resist against like viruses that are in South America. Transport it over into Africa, it's like getting infected by a whole bunch of new things. It doesn't really work very well. So that seems to be the common theme here. So with tomato and maize, it's the same kind of thing, okay? So, well, you won't find this in any textbooks. This is really my take on it. But I think we have this is really the encounter between, you know, some particular plant. We've moved it from one continent to another, and it encounters new viruses. Okay. So that's kind of relationships with, so okay, somehow the virus transmitted via vectors will arrive and be transmitted to a new plant. Yeah, I'll leave that for a question. So gets in, and the virus gets into the plant, and initially it will infect one cell. Oh no, it's my Google Translate infected cell. I didn't pick that one up, sorry. So c'est cellule infectée. So all plant viruses have local spread from cell to cell through the plasmodesmata. Okay, so these are the holes that connect the cytoplasm of neighboring cells in a plant tissue. And this is done by moving the plant genome from one cell to another via the actions of a movement protein. And there's basically two types of movement proteins. So either they will bind to the virus DNA or RNA and transport it through the plasmodesmata. And in that case, the structure of the plasmodesmata will stay the same, won't be modified. Some plant viruses have movement proteins that do something else. So these ones will radically change the structure of the plasmodesmata and increase the size, and this will allow the transport of a whole capsid from one plant cell to the next. Okay, so cowpea mosaic virus does this, and the TSWV does this. But one way or another, every plant virus has to do this. It's an obligatory step. For systemic transmission. So this will allow the virus to be transmitted within the cells of a whole leaf, for example. But it won't allow systemic transmission to the rest of the plant. In order for that to happen, it's got to be transported through the plant vascular system and stay infectious. And in that case, it's encapsulated particles which have this property. So systemic transmission to the whole of the rest of the plant requires the capsid protein. In almost every case. Okay, so let's see what you remembered here. And we just have to do this on a show of hands. Everybody ready? No DNA viruses infect plants. Who says yes? Who says no? It's a no. People have been paying attention. No double-stranded DNA viruses. Yeah, so this is not true. No envelope viruses infect plants. Who says true? Who says false? Yeah, you're right, it's false. So in the diagrams up there, there were some plant viruses that had an envelope on there. All plant viruses require vector for transmission. Who says yes? Who said no? The no people, you're right. This is the majority, okay? But not all of them, okay? The general case, yes, but there are some which do not. And D, you'll have to guess this because I didn't say it. But do you understand the difference or the two possible cases here? Oh, I wonder if I could, I don't have a green pen, but just imagine we got some kind of plant here like this. We got another one. Okay, that's some kind of plant. This one is infected. And we got a vector that's going to come. Somehow it's going to eat some of this plant or suck up some of the sap, and then it's going to go and infect this plant. So the question is, when the virus is here, is it actually infecting cells and replicating in the vector, or is it just being carried? You know, it's passive transport. Okay. So, I don't know. So for D, if you had to guess, what would you guess? That some plant viruses can replicate in the vector? You would guess yes. Anybody say no? Oh, said no. Great. Well, the answer is actually true. Some plant viruses can do this. But I find it very surprising because, you know, plant cells, insect cells, they're very different beasts. Okay, so their biology is very, very different. And somehow there are plant viruses that can manage to infect both types of cells and replicate efficiently. Okay, and that is the case. So if we go back to the table here, I believe, you know, these ones, the Bemisia tabaci, the virus, these Gemini viruses, they don't replicate inside the vector. They only replicate in the plant. So these vectors, you know, they're just transferring passively. So the vector will maybe not stay infectious for very long. Okay. Whereas TSWV, this one will actually replicate in the plant and in the vector. So it's got this like general capacity to be able to replicate in lots of different types of cells. So maybe that's also related to why it's not particularly fussy about its host species. So vectors, okay, sometimes the virus replicates, sometimes it doesn't. Okay, so let's have a look at our specific examples. So the first one is everybody's favorite plant virus, tobacco mosaic virus. So it's got a very, very simple non-enveloped helical capsid. You've got the plus strand RNA genome in the middle, and it's surrounded by apparently exactly 2,130 copies of the capsid protein. Now, when you look at the genome, got 6.5 kilobases of plus strand RNA. So the 5' end has a cap, and the 3' end has some secondary structures, doesn't have poly A. And you have 1, 2, 3, 4 open reading frames in the genome. ORF1 and ORF2 code for the RNA-dependent RNA polymerase. ORF4, that codes for the capsid protein, and ORF3 codes for the movement protein. So if you remember right at the beginning, what's the absolute minimum for a virus genome? It's one gene that helps it to replicate and one gene coding for a capsid protein. Plant viruses, the minimum is 3, okay? Something to help you replicate, capsid protein, and a movement protein. And that's it, that's all it's got, okay? So it's got the bare minimum. RNA-dependent RNA polymerase, a movement protein, and a capsid protein. That's it. Okay, so how does this guy replicate? So the vector will introduce whole virus particles into a plant cell. Now, apparently the 5' cap appears to be accessible because the first thing that's going to happen is that the ribosome will bind to the 5' end of the virus RNA and start to translate it. And as it goes along, it's going to knock off the capsid protein. So you get partial decapsidation of the genome. Then the proteins that have been expressed from this form, from this 5' end of the virus genome are what? The RNA polymerase, right? So you get the RNA polymerase which is expressed, and then it's going to bind at this end. I'm missing an S here. And so the RNA polymerase will bind here and start copying the plus strand RNA to a negative strand, and as it goes along, it will remove all of these ones. And then once that happens, then the RNA can be fully copied, and the shorter messenger RNAs coded for the movement protein and the capsid protein can be expressed. So then you get like full decapsidation and replication of the virus RNA, and then when the capsid protein is sufficiently sufficiently high concentration is present, then these copies of genomic RNA will start to be encapsulated with the formation of the helical nuclear capsid at the packaging site, the Psi sequence. Okay, so it goes through these cycles and you get new virus particles formed. So the other thing that can happen is that this new copies of genomic RNA can be complex with the movement protein, and they're transferred to a neighboring cell. Okay, so if you just had this happening and no movement protein, then these viruses will not be able to get out of the initial cell that was infected. Okay, so you just have a buildup of virus particles in one cell, and that would not really be very efficient. So we've known about TMWV for a very long time and understood how it works, and it's something that's been exploited to try and develop recombinant viruses for protein expression in plants. So there's two different recombinant vectors that are shown here. So in this one, we've got the full genome of the TMV, and we've just added green fluorescent protein in the middle. Where in this second one, the green fluorescent protein takes the place of the capsid protein. So what you can do is introduce these RNAs into the plant by agroinfection. Does this mean anything to you? So I didn't really put this in, so. Anybody heard of this guy? Okay, so it's gram-negative bacterium, which can infect lots of plants, and it's useful in plant biotechnology because it's got a plasmid, I think it's called the TY plasmid, and it forms a, let me get this right, yes, like a type 4 conjugation pilus, and it will go in and infect, so this is our bacterium, and this is our plant cell. And it would inject the plasmid DNA into the plant cell. So you get the plasmid DNA and it goes into the nucleus here. And then the genes that you've put on this plasmid will start to be expressed by the plant. So I think that's what they're doing here, okay? So they've, because you might think, yeah, how do they get this RNA into the plant? It's not easy, right? So that's what this agroinfection is. So you get this bacterium that's got the plasmid that codes for all of this stuff, and you can just inject it into the leaf of the plant, and that's enough. It will start the first cells where the TY plasmid that's transferred the plasmid DNA will start these plant cells expressing these messenger RNAs. Okay, so what happens here? So here's the experiment. So, and we got the analysis of green fluorescent protein expression, 5 days, 8 days, and what is that? 9 days post-infection. So here's the injection site on the 2 leaves. Okay, and you can see that the JL24, it's got a whole genome. So you inject it in this leaf, and then after like 8 days and 9 days later, you've got this first leaf is infected, and then you get a systemic infection through the rest of the plant, because the capsid protein is present, right? So it can cause a systemic infection. Whereas with this one, it's got the RNA polymerase, and it's got the movement protein. So it can infect neighboring cells and slowly manage to infect all the cells in this leaf, but doesn't get transmitted to the rest of the plant. Okay, so this type of vector has been developed for producing recombinant proteins in a plant. So you don't need cell culture or a bioreactor or something like that. You just get a whole load of plants lined up and infect them with your agrobacterium, and they will, the injected plants will start producing the recombinant protein. So you might say, well, this one is going to be better, right? Because then we get the infection in the whole plant, we get a bigger yield. But what would the advantage be of this one? Okay, let me give you a question. So could this virus ever escape from the greenhouse? Yeah, it could, because it's got a whole genome that could be functional. So conceivably, this one could somehow be transmitted to another plant. But that's not true for this one, okay? Because it's only going to be able to have local transmission. It would never really be able to survive in the wild. So from a biosecurity point of view, then this one will be better, okay? So, actually, I don't know if there are recombinant proteins that, yeah. Yeah, it's true. It is also quicker, but I don't really know what the explanation is for that. But it is, yeah. So knowledge of plant viruses and their molecular biology has allowed the development of these vectors for RNA viruses and also Gemini virus vectors. Okay, so tobacco mosaic virus, it's not a particular problem for us, because there's no tobacco being grown in France. But there are some related viruses that cause significant problems for farmers. So this one, the tomato brown rugose fruit virus, is also a tobamovirus. Similar structure, exactly the same kind of genome and replication cycle, was unknown until about 10 years ago, spread around the Mediterranean, and we now still have it as ever since now 2020. Infects tomato plants and peppers, and there doesn't seem to be any resistant cultivar. So the only way to try and prevent this virus from being transmitted and causing a lot of damage is anytime you've got some infected plants, then everything in that whole production unit has got to be destroyed. And I believe this one is transmitted by seeds. Okay, it's not particularly vector transmitted. So these viruses are a real problem in some cases. So another virus that causes problems in the northern hemisphere is the Bimovirus hordeoluteum, barley yellow mosaic virus. Once again, it's got a helical non-enveloped capsid with a plus strand RNA genome in there. The interesting thing about it here is that it's one of the plant viruses that has a fungal vector, which is the mold Polymyxa graminis. So the hyphae of Polymyxa graminis, when the spores germinate, they lay dormant in the ground. But when the spores germinate, the fungus starts to form hyphae, which will penetrate the root tissue of the plants that are growing there. And so this is the kind of like a section of a plant root, and this like amorphous blue stuff here, these are the hyphae of the Polymyxa graminis. So they kind of like really infiltrate into the root, and this is how the virus can get into these plants. So the real problem here is that the vector spores can remain dormant for 5 to 10 years, and the virus capsids that are associated with the spores can stay infectious also during that time. So it's a rare example, an unusual example of where a virus can persist without active replication in a host. Okay, so for almost all other cases, for plant viruses to persist, in a region, you've got to have continuous transmission cycles. Infected plant, the vector, and it infects another plant. And then if it gets to another vector, then it gets to another plant. And if you can interrupt the transmission cycle, your virus infection will disappear. But that's not true for this one, because the field stays infectious for 5 to 10 years. So what that gives you is something like this. So if you've got an infected field, what will happen is when the barley is sown and starts to grow in spring, you have most of the field here is looking okay, but you have these big yellow spots, and that's where the soil has been contaminated with the polymyxograminase and carrying the virus. So when you look at the plants here, they've got this kind of like yellow chlorotic appearance, and these barley shoots will not give a very good yield of grain. So if the field is really heavily contaminated, you can lose up to like 30 or 40% of the yield from a heavily contaminated field. So what's the solution for this for the farmers? Well, the only solution is grow something else for about 10 years, but that's not so easy for the farmers to do. And there are resistant cultivars that have apparently been developed, but I'm not sure they are used in Europe, been developed in Japan. Okay, what does the genome of these viruses look like? The barley yellow mosaic virus is a member of the Viridae family. And these are all plus-strand RNA viruses that code for a single polyprotein, and they've got a virus protein, VPG, at the 5' end of the genomic RNA. And when you look at the polyprotein, it codes for a couple of different proteases, RNA polymerase down here, the capsid is on the 3' end, and the VPG is in here. And the LNA-NLA protease will cleave most of these individual proteins here. Okay, every plant virus requires a movement protein, and for the potiviruses, it's this CI protein, or CI protein. Now, you might have picked up on this thing here, polyprotein and VPG. What does that look like? Well, it looks like a picornavirus. Okay, picornaviruses have got a very similar genome organization. The RNA has got 5' VPG, doesn't have a 5' cap. And if you look at the overall gene organization, it's kind of similar. So this one, the RNA-dependent RNA polymerase, they're homologous between these virus families. The NLA protease in potiviruses is homologous to the 3C protease in picornaviruses. And actually the C1, the movement protein, is homologous to the non-structural 2C protein in picornaviruses. So this part of the picornavirus genome looks a little bit like this part of the potivirus genome. So it kind of looks like, but the capsids are very different. So it kind of looks like somewhere along the line, a picornavirus has lost all the 5' end of its genome, okay? Loses all the capsid proteins. And it picks up something else, and it just sticks on the other end. So it's a different capsid protein coming from somewhere completely different, and that's why you get a different looking particle with no homology between the potivirus capsid protein and the capsid proteins of picornaviruses. So, and these kind of similarities are what allows, the ICTV and the people interested in virus phylogeny to group these potivirus and picornavirus families in the same phylum of plus-strand RNA viruses. Okay, so what about plant DNA viruses? So the only ones are the Gemini viridae, and they're called that because of the very specific capsid morphology in electron microscopy. So Gemini viruses have these very small kind of twinned icosahedral capsids. So it's very specific, and no other virus has got this kind of capsid structure. So if you look at these in more detail, it's like two very small icosahedral capsids kind of stuck together. And inside there, you've got the virus genome, which is single-stranded DNA. Now, there are 4 genera of Gemini viruses, and which are distinguished because of the genome organization. So the simplest, and if you look at the size of these circular single-stranded DNA molecules, they're all very small, 2.7, 3 kilobase, 2.8, and these ones are also, less than 3 kilobases in length. So one of the reasons is these particles are really, really tiny. So you can't get very much more DNA than that in there. And the other reason is because the movement protein of these Gemini viruses can't really complexes of more of single-stranded DNA and the movement protein more than 5 kilobases in length cannot get across the plasmodesmata. And the optimal size for DNA transport for these viruses is about 2.5 to 3 kilobases. So there's two constraints on the genome size. There's the size of the particle, and there's also the ability of the DNA to get from one plant cell to the next, which seems to be like very strongly constrained to less than 5 kilobases in length. Okay, so what other proteins coded here? Okay, so if we look at the Mastry virus genus, named after the maize streak virus, the first member. So this is kind of easy to see. We've got REP. This is a protein involved in the DNA replication. CP, the capsid protein, and MP, the movement protein. And then some auxiliary protein helps for DNA replication. And also for the Kurto and the Topoku viruses, we've got movement protein, capsid protein, REP, and some different auxiliary proteins. So all of them, they've got these three functions, okay? REP, replication, cap for the capsid, and MP for the movement protein. Now, Bergamo viruses, they're an interesting case because, and I'm going to have to like move up really close here because, so they've got the genome spread on two different DNA molecules. So DNA A and DNA B. And in DNA A, you've got the capsid protein and the REP protein. We don't have a movement protein on here. DNA B has got two open reading frames, which are involved in movement of the virus genome. So the NSP is a nuclear shuttle protein, takes the genome out of the nucleus and into the cytoplasm, and BC1 is the movement protein, which will transfer it to the next cell. So neither one of these molecules of DNA can replicate on its own, okay? They both need to be present in an infected cell in order for the virus to replicate effectively. Okay, so how does a replication cycle work? So after infection, you get whole capsids that enter into the first infected plant cell. They'll be imported into the nucleus. Decapsidation occurs. I'm not entirely sure, either during nuclear entry or slightly afterwards. So one way or another, you get single-stranded DNA released into the infected plant nucleus. So it appears that the host cell DNA repair mechanisms will fill this in and give you a double-stranded DNA copy. Once you've got the double-stranded DNA, then the host RNA polymerase can start transcribing messenger RNAs from the virus double-stranded DNA. And of these, you'll have like the REP complex. So the REP complex will bind to the origin of replication on the double-stranded DNA genome, and that's what will attract the host cell DNA polymerase and all the different cofactors required for DNA replication to the virus genome. Say, okay, to get entrance to the nucleus, REP initiates DNA replication, and then eventually the host cell DNA polymerase will go all the way around, and it will give you the initial double-stranded DNA and a new single-stranded DNA copy. And then the rep protein will also be responsible for detaching the copy from the original template to give you double-stranded DNA and a new copy. And then these cycles just repeat. Okay, so you get more and more copies of single-stranded DNA. So this virus DNA, two things can happen to it. It can be incorporated into a capsid, so can be disseminated through the vascular system of the plant and give a systemic infection. Can also be transferred from one infected plant to the other. Wow, I don't know, hang on, this is a very, very strange thing. Got a kind of invisible line here. It's just so weird. Okay. So all the DNA can be bound by the movement protein and transferred across into the neighboring cell. Again, you need both of these things to occur in order to get an effective infection. So Gemini viruses, they're the only DNA viruses that exist in plants. And the way the genome replicates looks a little bit, so this is what you would call what, it's kind of like a rolling circle replication mechanism, something like that. And some plasmids have the similar kind of mechanism of replication. So, and once you look, the rep protein also seems to be related to some proteins that are encoded in bacterial plasmids that facilitate the plasmid replication inside the bacteria. So there seems to be a phylogenetic relationship between the rep proteins of all these Gemini viruses and the equivalent that are encoded on plasmids that are found where? In phytoplasma. So phytoplasma are bacteria which are parasitic, they infect plants and they grow in the vascular tissue, in the phloem of plants. So the Gemini virus genome, apparently, some people, this is the theory, is it originated basically from a plasmid carried by bacteria that are also infecting the infecting plants. So you've got bacterium infecting plants, and somehow its plasmid DNA acquires the ability to replicate inside the plant nucleus. So that's nice for the plant. Nice for the Gemini virus, not so nice for the plant. But so you've got like the first part, the replication system, needs to have a capsid, right? So where did the capsid come from? So structural homology of the Gemini virus capsid proteins, they're all very, very similar. And the only similar type of capsid protein is found in an RNA satellite virus. So it seems like the Gemini viruses had an origin from a kind of really unusual encounter between a bacterial plasmid, somehow gains the capacity to replicate autonomously in the infected plant cell nucleus, and somehow this plasmid integrates the DNA coding for a capsid protein from a satellite virus. So that's apparently, what we think is how these viruses originated. Okay, so let's see if what you followed for the Gemini viruses. Yeah, and I showed up the Okay, so proposition A, the rep protein codes for DNA polymerase. Who says yes? One person. Who said no? Oh, to a couple of people. Who says no? The answer is no. It's too small. DNA polymerase needs more coding space. And these genomes are really tiny. A rep is maybe about 1 kilobase of DNA. So it's only going to be about 300, 350 amino acids in that protein. Too small. The function of rep, it's an origin binding protein, and it's what directs the host cell DNA polymerase to replicate the virus DNA. Now, what was that similar to in DNA viruses in animals? Small DNA viruses, circular genome. Might be like a transposer, but that's not what I'm thinking of. In small double-stranded DNA viruses infects mammals. Some infect primates and humans. The small DNA virus is a very important model system. SV40. And the origin binding protein of all these polymer viruses is the T antigen. Okay, so there's no homology between the large T antigen and the rep protein in Gemini viruses, but it's doing the same job. Okay, it's the origin binding protein, and it initiates the replication of the virus DNA by recruiting the host cell, RPA protein, primase, DNA polymerase, all the cofactors involved in DNA replication. So rep, not a DNA polymerase. It's an origin binding protein. B, the movement protein allows the entire virus particle to be transmitted from one cell to another. Who says yes? Who says no? Yeah, the answer is no. Binds a single-stranded DNA, and that's what moves. Gemini viruses are all transmitted by insect vectors. You have to guess. What would you guess? Who guesses yes? Who guesses no? I think yes, as far as we know, they are all transmitted by insect vectors. And what about D? D, okay, this has to be a guess. This is, you can figure this one out. If you look, so who says, yeah, that's true? Nobody. Who says, no, that's not true? Couple of people, most people are not really giving an opinion one way or the other. Okay, so the reason I asked this question is, okay, DNA A. That's why I put these up here, okay? So you can see DNA A, it's got rep, it's got the capsid. So it can replicate its own DNA, and this DNA can be put in the capsid. What is missing? The movement protein, no movement protein. So this can just, so it can form an infectious particle, right? Because you can have the DNA inside the capsid. But it won't be able to like get out from the initial infected cell. It will just stay there in the first cell that was infected. It can't really get out and infect the neighboring cells. So it can't spread within, well, maybe this is poorly worded question, so it's not obvious what I meant here, but the idea is that, this one can replicate on its own, but it can't really get transferred to the neighboring cell. And this one, DNA B, on its own can't do anything because it's got no rep protein. So it's just going to sit here as one single copy. Whereas DNA A, it's like just missing one function. Okay, so the begomoviruses are, what I hope anyway, are a good introduction to the next theme, which is the multipartite genomes. So the begomoviruses, their genome is spread on 2 different DNA molecules. And we've seen something like that with influenza already. So in influenza, the genome is carried on 8 different RNA segments with basically one gene coded for each segment. But so influenza virus is an example of a segmented genome. But in this case, every infectious particle carries one copy of each segment. So when you get infected by one virus particle, it's got the whole genome inside one particle. Begomoviruses don't work like that. In a begomovirus, DNA A is going to be in one particle and DNA B is in the other particle. And many plant viruses have this kind of organization. So for segmented genomes, the problem is, how do you incorporate one copy of each segment into the same particle? And for influenza virus, it's the RNA-RNA interactions that are specific between the different genome segments. Whereas for multipartite genomes, the problem is, how do we infect a host cell with all the necessary genes? Because in order for that to happen, several particles have to infect the same cell at the same time, so that you can ensure that you get some of these guys and some of these guys infecting the same cell. Now, if we were doing this in cell culture in a controlled system, what would we call, or what would be the term to describe this type of infection where you get several particles infecting the same cell? Anybody guess what I'm trying to get at here? What is the thing that we saw in the TD describes how many virus particles per cell? Nope. A quantitative term. How many particles? Yeah. Yes, MOI. So that means for these viruses to replicate, when they get transmitted from one plant to another, the infection has to be what? High MOI or low MOI? Yeah, it's got to be high, because you've got to have lots of the same virus particles going into the same cell. Why does this happen in plant viruses? Because they're very often transmitted by vectors. So especially, if you've got an insect that is like sucking up a load of sap from one infected plant, it's got, I don't know, millions of virus particles per microliter. And then it's going to go and inject those virus particles into the next host plant. So the infectious dose is really, really high. Okay? So I think the vector transmission is what allows this to happen. Because from one infected plant to the next, you get a lot of virus transmitted. That's not the case for influenza, right? So if I've got an influenza virus infection right now and I sneeze, I'm going to generate a huge cloud of droplets. But each droplet will have maybe like one virus particle in it. It's going to be dispersed over a wide volume. And then when you get infected by one particle. Okay, you have a low MOI infection. Okay, so I think that's the reason why we can see in several different virus families in plants this kind of multipartite genome idea. So it's easiest to spot in viruses with helical nucleocapsids, because when you purify viruses from infected plants, then you get a mixture of different capsid sizes. Okay, you've got this one with a long RNA molecule, and these ones will have short RNA molecule. And they'll have two different parts of the genome on the two different types of particle. So you also have this in some icosahedral virus particles, and you also have this in, like as I said before, the begomovirus genus of the Gemini viruses. Okay, so that's something we find in plants we don't find in animals. I don't think any animal virus has a multipartite genome. So I think the ability of vectors, like insect vectors, to transmit a high infectious dose at each round of virus transmission is also related to why you find a lot of these, what I've called capsidless genomes in plant viruses. So plant viruses can often be kind of associated with the replication of defective viruses. So if you've got an infected plant and you purify the RNA or the DNA from that infected plant, you'll find some complete genomes, but you'll maybe find a lot of defective virus genomes, which are dependent on the helper virus for their own replication. So these have been like viruses which have got incorporated a deletion mutant. So they've deleted some of their genome, but the RNA polymerase will still be able to replicate that genome. It can still be encapsulated in the virus particle. So if you're replicating a lot of RNA or DNA molecules, the ones that are going to be replicated faster are the shorter ones, right? Smaller ones. So there's a selective advantage for these deletion mutants, as long as they can rely on some of the helper virus to have the capsid protein and the RNA polymerase. And if you have a high MOI infection several times, then that's what you get. You get a mix of fully competent whole genome and then a whole bunch of defective viruses that are associated with it. So that's one of the reasons why, if you're maintaining a virus in culture, the passages you do should be low MOI, because then you get one infectious particle infecting a host cell. The only ones that can replicate are the ones that have got a complete genome. If you passage a virus with higher MOI several times, you get a whole bunch of defective viruses. So the other things that can that seem to be dependent on helper viruses are these umbra viruses and satellites, which I'll talk about briefly. So umbra viruses, they are almost independent. Okay, so these are, 4 kilobases of RNA plus strand RNA, coding for 4 open reading frames. So these two, they code for the RNA polymerase of the virus, and that's what's going to make the intermediate negative strand copy. They're at the 5' end, so the host cell ribosome is going to translate these guys first. Okay, so it's looking a little bit like our tobacco mosaic virus genome. Okay, it had the RNA polymerase at the 5' end. You get a negative strand replicative intermediate, and this negative strand copy is the template for full-length genome copies and a shorter messenger RNA that will be coding for these other 2 open reading frames. One of them is the movement protein, which can take this one and allow the virus to infect the neighboring cells. And 3 is, the open reading frame 3 codes for a dissemination protein, which allows the virus to infect the rest of the plant through the vascular system. So umbra viruses, they are an exception. You can have systemic infection without a capsid protein. So as I said, they're almost independent. So if this genomic RNA gets inside a plant cell, it can infect the whole plant. But it can't move outside of that plant because it's got no capsid protein. So it can't survive if it's being eaten by an insect vector. Okay, so it's only dependent on the helper virus for encapsulation of the genome and transmission from one plant to another. And the helper virus is, I believe, a luteovirus. I think it was on the previous slide. So umbra viruses, they're almost independent, but they require a co-infection by another virus in order to be transmitted from one plant to another. Now, satellites, they're a bit more dependent because they don't have any RNA replication at all in the absence of a helper virus. Some of them can code for proteins, And when they co-infect a plant, they'll modify the symptoms. Often the symptoms will be attenuated by co-infection with a satellite. So there's a distinction between satellite viruses and satellite RNAs. So satellite viruses code for a capsid protein, and the capsid protein will, incorporate the satellite RNA. So it can be transmitted from one plant to another, but it can't replicate. It's kind of like the opposite of the Umbra viruses. So, and as well as that, there are satellite RNAs. They don't code for a capsid protein. Some of them don't code for any proteins at all. Some do, and in which case, the function of the protein will be to direct the helper virus RNA polymerase to the genome of the satellite. Okay, so these are kind of like parasitic RNA molecules that are dependent on the helper virus. And they don't have any real replicative capacity of their own. And the last example are the viroids, which are a different kind of beast altogether. So they are independent. They don't require a co-infection from a helper virus, and none of them code for proteins. Oh, I didn't change this. Okay, so there are two families of viroids, the Puspii-Viroidae. So whenever you've got a viroid, instead of having V at the end of its name, it's called VD. So this is potato spindle tuber viroid. And it's the same kind of convention for names as for plant viruses. So the first one, the host plant, and then something to do with the symptoms. And the other family are the Avsun viroids, which are, I can't remember, ASBVD, so it's avocado something something viroid. I wonder if I have that in my notes here. I didn't write it down. Oh, okay. So these are just single-stranded circular RNA molecules with extensive secondary structure. So the RNAs will have extensive base pairing with themselves. Okay. So the circular RNA is not like a big circle, it's more like quite a rigid like rod of RNA. And for some reason, which I just do not understand at all, the molecular process here, but it's the host cell RNA polymerase II, which takes this as a template and makes another RNA copy. So they replicate either in the nucleus, that's for the Puspii viroids, or in the chloroplast, that's the Avsun viroids. And so they were discovered because they cause symptoms in infected plants and it's transmissible. So the first one was the potato spindle tuber viroid. So these are potatoes from a non-infected plant, and these are the potatoes from an infected plant. So you can see they've got like the form a bit like, it's a bit elongated, like a fusiform sort of thing, like a spindle. So they are able to get a systemic infection, so they can be transmitted from cell to cell and through the plant vascular system, and is dependent on the host cell RNA binding proteins, which will bind to the viroid RNA and move it around from cell to cell and through the whole plant. So you kind of wonder, how do these things cause symptoms? Well, it's just an RNA molecule getting replicated. Now, why does this affect the plant biology at all? Well, it appears that the viroid during replication also results in the production of short interfering RNAs, siRNAs, which can have an effect on the expression of host genes. Okay, so some of these viroid-derived siRNAs are interacting with host gene expression, and that's what will cause the symptoms. Okay. So plants don't take this lying down. They have mechanisms to try and destroy intruder RNA. So these are the dicer-like proteins, which are apparently involved in the resistance to viroid infections. Okay, so that's about the end for today. So main things to remember in plant viruses: unusual characteristics of their genomes, no double-stranded DNA viruses, and they have these weird subviral forms like the satellites and the viroids and umbra viruses. I think, honestly, we find these in plants, not in animal viruses, probably related to their dependence on vectors and the high MOI at each transmission cycle. That's what allows these things to persist. In plant virus pathology, every plant virus needs a matrix movement protein. Geez, I always get this wrong. Movement protein for cell-to-cell transmission, and for systemic transmission, it requires an encapsulated virus particle. Yeah, that's what already kind of talked about this. And maybe the last thing is, it seems that the appearance of new strongly phytopathogenic viruses seems to be related to the encounter of a new virus to a new host plant, and whether this virus infection can spread or not really is dependent on whether the vector covers a large geographic region or is restricted. And so as insect vectors spread, they'll bring plant viruses along with them. Okay, so that's it for plant viruses. Last lecture next week, which I still have to finish, is on mostly human infections, and the last bit I need to do is to have a couple of slides on prions, which are the kind of opposite of viroids, and you'll see what I mean by that next week. Okay, thanks very much, everybody.